Hi, I'm Jesus Pena, and I'm a microbiology PhD candidate in the STICH lab at the University of California, Riverside. I'm originally from Denver, Colorado, and I moved here to study the evolution of sexual reproduction in fungi. I'm especially interested in the mechanisms that the zygomycete fungi have evolved to determine mating identity and what genes play a direct role in the process of sexual reproduction. I chose UCR because I wanted an environment where I would be exposed to diverse subdisciplines outside of my own immediate interests to ensure my own intellectual development. The opportunity for interdisciplinary research in the microbiology program has actually helped me to gain new perspective on my own research. We're standing outside of the genomics building, which is home to many labs in the microbiology program, including my own. The range of research that happens in this building varies from nematology and fungal biology to virology and plant genetics. Let's go check it out. Welcome to the STICH lab. We do a lot of genomic work here, so our benches have a lot of the standard tools of molecular biology. When extracting DNA or RNA from fungi, we freeze them in liquid nitrogen and place them in this mortar. Then we crush that tissue down into a fine powder, further process it with a bunch of chemicals to purify the nucleic acid that we're interested in before sending it off for sequencing. UCR has its own sequencing facility, which is pretty convenient for amplicon sequencing, but there are other options for whole genome sequencing as well. In fact, my lab is working on getting a new sequencing system going. Let me show you. Over here is our nanopore sequencer, a revolutionary technology that is capable of sequencing genomes in the comfort of your own lab. My lab is trying to get this system up and running so that we can sequence way more fungal genomes. We've already tried it on bacteria and plant genomes, collaborating with other labs here at UCR. Over the span of a couple days, this little device will actually spit out the DNA sequence of whatever organism you might be interested in. You may have also noticed behind me the microscope, and you may be wondering why a genomics lab has this type of equipment. So we're not just DNA sequencers, we also do quite a bit of culturing. Just another example of the interdisciplinary work that happens here at UCR. Let me show you the culture room. This is our culture room. This is where we keep all of our living organisms. The majority of our collection comes from the US Department of Agriculture, as well as samples we've collected in the field, and even UCR's herbarium. This is my organism of choice, Phycomyces blakesleyanus. It is the fungus that can see the stars. Now, what that means is that it can actually sense light. It can detect light as faint as starlight. We've also got black yeasts that were collected from Joshua Tree National Park. We've even got some lichens somewhere that have been collected from all over Southern California. Well, I hope you enjoyed this tour of the STICH lab at UC Riverside in the Department of Microbiology and Plant Pathology. We hope to hear from you soon, so fill out an interest form. You can also follow us on Twitter.